Welcome everyone today to this joint webinar series between GlycoNet and the ACS carbohydrate division. And it's my pleasure to introduce our first speaker for today, who is Professor Kelly McNagny, who is from the University of British Columbia. Kelly describes himself as a stem cell biologist, and he's a professor in the Department of Medical Genetics, and he's co-director of the Biomedical Research Center at the University of BC. Dr. McNagny got his PhD from the University of Alabama at Birmingham, where he worked with Max Cooper at the Howard Hughes Medical Institute as part of the National Academy of Sciences. He took a postdoc in Europe, where he was at the European Molecular Biology Labs in Heidelberg. I did mine in Freiburg around the same time, so we were there at the same time, Kelly, where he worked with Thomas Graf, and his work there was on transcriptional control of hematopoietic stem cell maturation. And he stayed on there for a little bit as a semi-independent scientist before taking his position at UBC. And so Kelly's been at UBC since 1998, I guess. And during that time, he's received several awards, including ones from CIHR and the Michael Smith Foundation, and also from the Shoal Pfizer Junior Faculty Award from the American Association of Immunology. Kelly's lab studies the CD34 family of sialomucins, something that those of us who are interested in sialic acid in biology, of course, are very interested in, and I look forward to hearing your talk today, Kelly.
Thanks very much, Warren. My pleasure. And thanks for the invite to you and to the network for being able to speak with you. Just as a quick outline, what I wanted to do today is very quickly cover some background information on the CD34 family of sialomucins, which is a family we've studied for well over 20 years now. I'll do that quickly because most of that data is published, but I just want to give you a flavor for some of the things we found with that family. And then I want to spend a bit more time on Podxl, one of the members of that family as a tumor target for immunotherapy, and tell you about it being a marker of poor outcome tumors consistently, that it plays a functional role in tumor progression and metastasis likely, and that it seems to be a good target for immunotherapeutic approaches to cancer treatment. So very quickly, this is the family of molecules that we studied. This is our family portrait. CD34, as most of you I'm sure know, is a classic sialomucin. It's really the oldest stem cell marker and consistently even used today in the clinics. There's over 40,000 publications on CD34 as a marker of stem cells. And yet really very little is known about its function. Very few of those publications address that. It's a classic sialomucin with lots of O-linked and some N-linked glycosylation, a globular domain, stalk domain, and then a short cytoplasmic tail of about 72 amino acids with a C-terminal PDZ docking site. Now, a number of years ago, Mac's lab knocked out CD34, and so did Lasky's group. And it was sponsored by a couple of big pharma companies trying to show a function for this molecule. And really, the phenotype of those mice was not much. The mice were completely happy, healthy, and normal with a few very minor defects. Right about that time, we cloned this second molecule called Podxl. And we immediately realized that it was a very closely related family member. It had all the same structural biochemical motifs, including the C-terminal PDZ domain docking site, had a similar genomic organization. It was clear the genes had a common ancestor. And at the same time, we also cloned a third family member based on sequence homology between these other two called endoglycan. And we've looked pretty extensively. We think this is the entire family. All of them have similar biochemical motifs. All of them are expressed by vascular endothelial or vascular associated tissue, and all of them are expressed by early hematopoietic progenitors during embryogenesis. So overlapping expression and overlapping biochemical motifs. In terms of things that bind to the intracellular domain of the molecules, we've done a bit of work on that as others have as well. We have found one PDZ domain scaffolding protein called NRF1 and NRF2, bind very effectively to the C-terminus of podoclixin and endoglycan, but not to CD34, likely because of some differences in the PDZ domain docking site. These proteins bind a who's who of signaling molecules, and things like tyrosine kinases, PDGF receptor, VEGF receptor. They also bind a slew of G protein coupled receptors. They bind beta-catenin, some signaling molecules. And we think that podoclixin and endoglycan determine where that scaffold goes, and presumably all those molecules that are bound to that scaffold. So it probably plays a role in signal transduction. Now, just very quickly summarizing about 20 years of work from my lab and others. In general, we think that these molecules play just a biophysical role in blocking adhesion, or they act like molecular Teflon. So all of them have a very highly sialylated extracellular domain, lots of negative charge, and they tend to commingle with integrins and other adhesion molecules on non-adherent cells. And we think that they act to just block non-specific adhesion when they're in that context. They can move very rapidly out of the way of those molecules when you activate cells. And so we think when you move them away from integrins and other adhesion molecules, you now allow those molecules to start working as pro-adhesive molecules. And in fact, these can actually facilitate the segregation of molecules to mutually exclusive domains, which likely explains a lot of confusion in the literature concerning whether these molecules act as Teflon or anti-adhesion molecules, or Velcro as pro-adhesive molecules. And I'd be happy to talk about that later. On adherent cells, epithelial cells, and other cell types, they also regulate the apical domain size of cells. So if you overexpress podoclixin, for example, in an epithelial cell line, you get a pretty striking apical domain expansion, and can even get beautiful sprouting of microvilli to cope with that extra apical domain on the apical surface of the cells. So they regulate topology in cells as well. In rare cases, and I want to stress rare cases, they can serve as pro-adhesive molecules. So the classic leukocyte trafficking into lymph nodes via L-selectin is mediated through CD34 and podoclixin and endoglycan. Each of those molecules is expressed on the endothelia, where they get a sialo-Lewis X modification. That allows them to be recognized by L-selectin, and that starts the rolling cascade, which allows them to taste the endothelia, become activated, and firmly adhere to the endothelia before diapedesing into the lymph node. But I want to stress that that's the exception rather than a rule. We can't find that appropriate glycosylation anywhere except on high endothelial venules in the lymph nodes. So we think they, in most other cases, serve as blockers of adhesion. And then the last thing I wanted to stress just for this talk, we also have some evidence that they serve as more specific roles in chemotactic signaling. For example, if you knock out CD34, you find that you tend to lose the ability of cells to migrate to certain chemokines, ligands for CCR3, CCR7. But those same cells are perfectly fine for migrating towards CXCR4 ligands, CXCL12. Conversely, if you knock out podoclixin, these cells migrate fine to a whole variety of chemokines, but they selectively lose the ability to chemotax towards CXCR4 ligands. And in my lab, we tend to think of mucins as either Teflon or Velcro, but if all they were is acting as Teflon or Velcro, you really would only need one molecule, and that wouldn't explain why you have hundreds of different sialomucins expressed in different patterns. We think that actually these tend to pair with subsets of chemokine receptors and act as rudders to guide the way cells migrate. And so some of them will bind to a subset of chemokine receptors, others will bind to other subsets of chemokine receptors, and they act as a code for helping you migrate to the right place in the right time. Okay, last couple of bits of summary. So when we recognized that this was a gene family and we realized there was overlapping expression in some tissues like hematopoietic progenitors and like vascular endothelia, we realized we probably wouldn't get a strong phenotype if you looked at a single family member knockout where you have all this functional redundancy. Where you would get a phenotype is where you found these proteins are expressed singly. And in the case of CD34, it's selectively expressed by these hematopoietic lineages, dendritic cell precursors, strongly expressed by mast cells, and eosinophil precursors. And we reasoned that you would have a strong phenotype in disease models where those cells were important. Conversely, podocolixin is selectively expressed on podocytes where the other family members are not. And we reasoned you would have a strong phenotype in that tissue for podocolixin knockouts. And that's essentially what we looked at next. So this is a busy slide. It shows a bunch of references from my lab. And essentially now, instead of having no phenotype with the 34 knockouts, when you looked selectively, you could find a phenotype almost everywhere you looked. So if you put them in inflammatory disease models that require mast cells or eosinophils or dendritic cells, you find that in every instance, the mice are resistant to those diseases. They get very little inflammation in response to allergic asthma, hypersensitivity pneumonitis, inflammatory bowel disease, all diseases where you require appropriate migration of these cell subsets. Conversely, you would also find that they were sensitive or more sensitive to some of these other diseases where we knew CD34 was expressed, but still required the migration of these cells in order to fight damage. So in muscle damage model, they're more sensitive. Same in an arthritis model where there's a defect in vascular endothelia. And for the podocolixin knockouts, again, same story. The hematopoietic progenitors seem to be relatively okay in the podocolixin knockouts. This is the endothelia. But you see a very potent defect in kidney function if you knock it out embryonically in mice, because it's required for kidney morphogenesis and also required for tissue movement and sliding around during embryogenesis. And the knockout mice tend to have some birth defects if you knock it out early in development. What I wanted to switch to today, though, is I wanted to tell you more about our work in tumor progression. Now, knowing that each of these molecules plays a role in facilitating migration and enhancing mobility, we realized cancer is a mobile and invasive disease, and we wanted to see if any of them were upregulated during tumor progression. And here's the first experiment we did. We took a case tissue microarray of about 300 breast cancer patients. This was from the BC Cancer Agency. And these were old cases. We had 20-year follow-up with them. And if you looked at the cohort in general, you find that the bulk of them had low expression of podocolixin. And if you looked at what happened to those patients over time, after 20 years, most of those patients were alive. And that's because if you catch breast cancer early and you take out the lump and you don't see local lymph node invasion, no evidence of metastases, you tend to send the patient home and say, have a good, happy, healthy, normal life. And most of those patients do quite well. On the other hand, there is a subset of those patients who will go on to relapse, and it's been very difficult to predict which ones will and which ones won't. Turns out that podocolixin was expressed at high levels by between 6 and 10% of those cases. And in every single case, those patients were dead by 15 years post-follow-up. So podocolixin is a really good predictor of patients who are going to relapse. And in fact, you're about 8-fold more likely to die from your breast tumor if you've got podocolixin on it at diagnosis than if you don't. And it's an independent predictor of bad outcome, independent of tumor size, independent of tumor grade, lymph node involvement at the local level when you do the first imaging. It's independent of HER2 overexpression, ER loss, p53 mutations. So it's really a good independent predictor of bad outcome disease. The same is true in ovarian cancer. Here's just a couple of examples. Here's some diffuse staining, both cytosolic and surface staining on ovarian cancers weekly. Here's strong expression as the disease progresses. If you look at the diffuse staining cancer patients, they did pretty poorly because ovarian's a bad disease when you finally catch it. But if you look at the podocolixin surface expressors, they did even worse, and that's really significant, quicker death if you have podocolixin on the free surface like you see here. That's our data, but since we showed that, there's been a cottage industry of people looking at podocolixin in a whole different variety of tumors. Here's an example with breast cancer done by a Swedish group. Again, no podocolixin on the tumor. The patients do reasonably well. High podocolixin on tumor, really bad outcome. Same is true in colon cancer. Here it's interesting. This was a study where they looked at over 300 cases of colon cancer. The Podxl negative cases did poorly, but if you had high levels of Podxl on tumor, you did really poorly. And interestingly enough, in colon cancer, there are a couple of treatments. You can either choose to have surgery and remove your tumor, or you can have surgery plus adjuvant therapy, chemotherapy. As it turned out, in these cases, half of the patients had adjuvant therapy and half of them didn't. If you look here at what happened to those patients, the ones that had high Podxl on their tumors and had chemotherapy did actually quite well. The ones that had Podxl on their tumor and had no chemotherapy did poorly. So this tells you that not only is it a predictor of bad outcome disease, but if you know that there's Podxl on your tumor and you get adjuvant therapy, you'll likely do much better than if you don't. Okay, a quick summary here of just all the cancers where people have looked at Podxl expression. You can see where the arrows are. There's a whole variety of different tumors that upregulate. In every single case, it correlates with bad outcome. Where you don't see an arrow doesn't mean that it is not expressed there. It just means it hasn't been published or studied yet. And I think it's going to turn out that Podxl is a predictor of bad outcome disease in just a whole variety of different tumor types. So that's good. We know we've got a nice diagnostic marker. We know that if you start using that at diagnosis, you might be able to change the course of a patient's disease by getting in and giving them aggressive therapy early. Is it playing a functional role? And the way we address that is, here's a simple experiment. We took MDA231. This is a really aggressive breast cancer cell line that causes great tumors in mice. Cells have very bad polarity. We took those cells and we labeled them either red or green. And in one case, we knocked down Podxl expression using an shRNA viral vector. We then mixed those cells together and popped them into mice, and then asked 6 weeks later, which cells made tumors? Was it both, or was it only the wild type or only the knockout? And briefly, for the first 2 weeks, actually, these are intravenous injections, there was essentially no difference. If you took the lungs from these mice and you looked at red and green cells, you found an equal number for the first 2 weeks. But if you looked 6 weeks later, it was really night and day. So in the case on the left, I'm showing you the lungs of these mice. They're absolutely riddled with tumor cells. And if you look, all of those tumor cells come from the wild type tumors. None of them come from the knockdown cells. It's not a case of a clone or something. If you flip the labels around, do the same knockdown. Now here, the wild type cells again give beautiful tumors with a control vector. With the Podxl knockdown vector, no tumors. So what that tells you is knocking down Podxl expression cripples the ability of these tumors to form in mice. The same is true, really, if you inject those cells subcutaneously. This shows cells that were injected in the flank of a mice with control vector. And you can see they shred right through the muscle layer. These are muscle fibers shown here in pink. If you do the same with the Podxl knockdown cells, the tumors stay relatively encapsulated. They don't shred the muscle. They don't burrow into the bone like the wild type tumors. And that was our early study. It's now been reproduced by Weinberg and his colleagues. He's seen essentially the same thing. Expression of Podxl really enhances the ability of cells to form tumors in xenografted mice. It's not just breast cancer cells. We've done the same thing with an ovarian cancer cell line, again, knocking down Podxl, or in this case, knocking it out using CRISPR. Really slows tumor growth dramatically. And here's a pancreatic cell line where we completely flatline tumors if you knock out Podxl. So it looks like in a whole variety of different tumor types, you can block the ability of them to form primary and metastatic tumors in particular if you knock down Podxl. Now, knowing that, and knowing that this is a sialomucin, and knowing that tumors and other cell types, in fact, tend to have tissue-specific glycosylation, we thought perhaps we could make some antibodies that recognize the tumor glycoform of Podxl, but don't react with any normal tissue. And those might be useful as therapeutics for cancer. So I want to tell you quickly about two antibodies. One is called POTO83. This one does not bind a glycoepitope. It binds the stalk domain of podoclixin right next to the membrane. It's a juxtamembrane epitope in the core protein. I wanted to show you that one because it gives us some insight into how blocking podoclixin could block tumor progression. And then I want to tell you a little bit more about a very tumor-specific glycoepitope antibody called POTO447, which recognizes an unusual beta-1 GalNAc epitope in the context of the protein backbone. Okay, so we made over 200 monoclonal antibodies against human podoclixin. And then at that stage, we were actually kind of stuck for how to screen them. Because if you knock out podoclixin in cells and grow them in a dish in 2D culture, you don't see a dramatic change in the phenotype of those cells. So we did a very heroic assay, I'm going to argue. We took those MDA231 cells, we coated them with some supernatant from the monoclonals, and then we popped those cells into mice and asked if any of the antibodies we had generated would slow tumor growth for the first couple of weeks in those mice. And here's the first 10 antibodies we tried. And I want to highlight this antibody here, POTO83, which really flatlined growth of tumors for the first 2 weeks. So that's looking good, seems to be blocking cells. You don't usually know you're going to get cancer before you get it. So we tried then to first let the tumors engraft in the mice. And we waited until the tumors were about 100 millimeters cubed. So that's about the size of a grain of rice. And then began treating with antibody. Here's a control antibody. The tumors grew unabated. Here again is that POTO83 antibody in an established tumor, flatlines tumor growth. So pretty striking, 4 injections, no tumor growth. Here's what the tumors look like. And in fact, I'm not really convinced this is a tumor. This is a matrigel plug that we pulled out of the mouse after we had treated with the antibody. And here's what you see with the control antibody. Okay, so that's with a small tumor. You know, it's big for the size of a mouse, but it's still a small tumor. What if we really push the system? So in this case, we waited 20 days till the tumors were 750 millimeters cubed. That's really close to when you're going to have to kill the mice because they've reached their humane endpoint. But we wait until that stage, then we again treat it with a control antibody or with the POTO83 antibody. And what you can see here is if you're an optimist, you can see that actually even in this size tumor, it looks like it's stemming the tide of those primary tumors on the flanks of the mice. But what was interesting is if you also looked at the metastases in these mice. And we know that the tumors from that primary will begin forming mets in the lung somewhere around day 14. So we haven't started treatment until after that process has already started. So when we looked at the lungs of those mice, here's what you see. So with the control antibody, you see the lungs are absolutely riddled with tumors, too many nodules to count. These are all glowing in green. But here's the mice that got the POTO83 antibody. And you can see, you can count small nodules in the lung, but it's night and day compared to the control antibody. Really, we've dramatically knocked down met formation in these mice by injecting the POTO83 antibody. Okay, so that's great. Looks pretty good. Looks like we've got something that even without any drug conjugate or anything at all as a naked antibody seems to block a functional epitope on podoclixin. The problem with that antibody is highlighted here. So it is exquisitely specific for human podoclixin, doesn't bind mouse podoclixin, doesn't even bind macaque podoclixin. So doing toxicology studies with that antibody is a bit of a challenge. I can't guarantee that if you injected that antibody into a patient, you wouldn't have a bad effect on endothelia or hematopoietic progenitors or podocytes in the kidney. I can give you some hand-waving reasons why we think that might not be so much of a problem. We could talk about that later if you're interested. But in order to do proper toxicology studies, what we've done now is we've cloned the mouse, we've cloned the human gene and knocked it into the mouse podoclixin locus. So now we have a humanized mouse where human podoclixin is expressed in all the normal tissues in mouse. And we're just starting to use those mice to test for toxicity with this antibody and are hoping that if we don't see any tox, we can now move on to some tumor studies. So that's with that antibody. But I also wanted to remind you that we generated a second antibody called POTO447, which is exquisitely specific for a tumor glycan on podocalyxin. So some of that data is shown here. Here are 3 different cancer cell lines: a glioblastoma cell line, a pancreatic cancer cell line, a breast cancer cell line. In blue, I'm showing you the POTO83 core protein epitope. And in red, I'm showing you the POTO447 reactivity. On the tumor cells, both are co-expressed, both epitopes are expressed. If you look on HUVEX, though, normal human umbilical cord blood endothelial cells, you find that the POTO83 epitope is expressed at high levels, but the POTO447 doesn't bind at all. So these normal cells don't express that glycoepitope. To show you that that antibody is indeed podocalyxin specific, though, we've knocked it out in 3 different cell lines. So here's the POTO83 reactivity. It's gone when you knock out podocalyxin using CRISPR in every case. Same is true for the 447 antibodies. So you've gotten rid of the epitope staining when you knock out the gene. And because these cells have not changed their glycosylation pattern, I think it's safe to say that there are plenty of proteins here that have, differences in glycosylation from normal cells, but we've lost reactivity with the POTO447 antibody. So it's recognizing a glycan in the context of podocalyxin's core backbone. Just some more flesh on the bone for that. In this case, we've treated the cells with either with neuraminidase to try to shave off some sialic acids. That doesn't tend to change the staining pattern with either antibody. But if you treat with endopeptidase, which cuts below the mucin domain and leaves just the core stalk protein behind, you tend to start losing the POTO447 epitope while you retain the 83 epitope. And we've also taken knockout cells and re-expressed human podocalyxin as either the full-length protein or just the stalk domain. You can see with the full-length protein, you get the 447 epitope back. With the stalk domain alone, you don't. So we think it's recognizing a glycoepitope in the mucin domain. Now, to convince you that that is truly tumor specific, we've done a whole bunch of staining on normal human tissue. This is podocalyxin core protein reactivity in kidney. This is beautiful staining on kidney podocytes and on vascular endothelia. Here's the glycospecific antibody, and you can see it doesn't stain those kidney podocytes which express podocalyxin at all, nor does it stain vascular endothelia. We've also looked at a whole bunch of normal human tissues and don't see any expression there. And it's not just us. We've actually collaborated with a CRO, Charles River, to look at normal tissue exposure. They can't find any normal tissue expression of that epitope. Of course, not all tumors will have that glycoepitope, only a subset will. We've focused our attention on ovarian cancer because if you look at ovarian cancer, virtually all of them express podocalyxin. But if you look at the POTO447 epitope on the cell surface, really about 64% of the ovarian cancers express that glycoepitope. So we think that would be a good indication for using this antibody as a therapy. The antibody doesn't have an effect on its own. It doesn't block tumor progression as a naked antibody. But if you conjugate it to make an antibody drug conjugate with vedotin and MMAE as a toxin, this is a toxin that needs to get within the cells and to kill them. We find that that conjugate is very effective at killing pancreatic tumor cell lines in vitro, whereas a control antibody has no effect. And if you do the same experiment with a knockout cell line, you get no killing of the knockout cells with either antibody, the control or the POTO447 antibody. Same is true for the ovarian cancer cell line. Okay, so that's cells in a dish. What about in vivo? Here's our NSG mice. These are, immunodeficient mice where we've stuck in this pancreatic cell line in. You can see that when you begin treatment with the POTO447 antibody or a control antibody, the control antibody gives you a slight dip with antibody treatment, but then the tumors rebound and grow well. Podocalyxin antibody 447 completely wipes out tumor growth. That's at 4 mg per kg, pretty similar to 2 mg per kg, and that's really industry standard treatment. If you do that experiment with the mice, the 447 treated mice all survive. All the ones that didn't get that antibody, got a control antibody, die. And we've done the same thing in nude mice just to convince you it's not something that, is tightly linked to lacking B cells and NK cells. And actually, in this case, you don't see that dip with the control antibody. And I think that's because the NSG mice have no T cells or B cells, which means they have no serum antibody. So you don't get any non-specific toxicity with nude mice because all of their surfaces are pre-coated with serum antibody. You don't see that dip, but you get the same effect with a Poto 447 antibody. It really blocks tumor growth. Lastly, we've also made that antibody into a single chain antibody, and we've grafted it onto an off-the-shelf CAR T vector. So if you graft that single chain onto a CAR T vector that has a CD8 hinge, a CD28 transmembrane domain and cytosolic domain, and CD3 zeta signaling chain, you get great expression in primary T cells, either CD4 T cells or CD8 T cells. If you take those CAR T cells and you mix them together with a glioblastoma cell line in a dish, the control vector does not kill the cells, but the POTO-CARs are really effective at killing cells in vitro. And you get essentially 100% killing if you get the right target ratio. So it could work as a CAR as well. So just as a quick summary, what I've tried to show you today is that Podxl is an interesting member of the CD34 family of stem cell sialomucins. It plays a key role in regulating cell adhesion and migration and invasion. It's consistently upregulated on tumors, and we can talk about it, but our data suggests that you really need it expressed by tumor-initiating cells or tumor stem cells in order for them to metastasize. And by removing it, you cripple the ability of those cells to form distal tumors. I've shown you that we have an antibody that blocks a functional epitope on Podxl and can work just like a knockout. And I've also shown you that we have a glycospecific antibody, exquisitely specific for tumor glycoform of Podxl, and that seems to work well as a antibody drug conjugate or as a targeting domain on a CAR T vector. And we think both of these antibodies could be very useful in either cell imaging or in treating cancer as a therapeutic. These are the people in my lab who've done all of this work. Julianne Brassard is a new member of my lab who's looking at hot and cold tumors. These are tumors that do or don't have lymphoid infiltrates to see if there's a correlation with Podxl expression. Diana Canals did all of the in vivo killing that I've shown you and has done all the preliminary work with a 447 antibody. And Mike Hughes has been involved with me from the very beginning of the Podxl studies in cancer. Close collaborator Cal Ross Kelly, and a new collaborator, Kevin Leslie, who's a terrific guy for immunotherapeutic studies and has been guiding a lot of our work there. And then the group in Copenhagen who's been helping us map the glycoepitope. Okay. Thank you.